Next, I will talk about several examples about cell therapy of the regenerative medicine field. In my experience, I have more experiences in mesenchymal stem cell product development. At the first, our team make mesenchymal stem cell banking from healthy donors. We isolated mesenchymal stem cell from bone marrow, from adipose tissue, and also from cord blood water jelly, like cord, water jelly of the cord. The source of the cell or tissue are fully screened through the process by pathogen, by other, and also some gene expression function, and also proliferation efficiency of the cells. After over 20 lines of cell screening, we identify several good candidates to further process in our GTP facility. Now we have processed these several lines of mesenchymal stem cell go to preclinical test, then go to clinical trial, and have 2 IND approval in Taiwan. The mesenchymal stem cell core technology, as I have mentioned previously, from the isolation of healthy donor to dedicated screening of their function, gene expression, and also proliferation efficiency. We using multiple molecular and serial testing platform to test the cell efficacy, the cell function, to make sure that when patient receive all these cells, will make best benefit. And the cell bank that will meet all clinical regulation requirements that international guidance have all mentioned about. And also the final product formula will meet the clinical using standard. We set up several kinds of formulation of the product to meet the clinical delivery process. The first example we set up to go to the clinical use of the mesenchymal cell bank is using this type of cell in diabetic wound healing, especially in the chronic wound healing of a lot of diabetic patients. We show here the animal study of the rat animal model of diabetic wound. As you can see, when we using mesenchymal stem cell that transplanted to the muscle tissue, we can see the healing process more efficiently, and the healing tissue have more integrated skin and muscle, and also have a high expression level of collagen expression. Then we also can see the inflammation effect of this wound is reduced by the cell therapy process. Another example of clinical use, what we have now in IND open condition, is the acute AMI myocardial infarction. And we using porcine model to test in the clinical condition. As shown here, we can see that when we transplant the cell into AMI porcine animal model, after several months, the heart function is significantly improved, and also the inflammation of the heart tissue is reduced. It's very significantly, and all the clinician of heart is very impressed. So now these 2 kinds of clinical application is now go to clinical in the phase 1 condition. After we have identified several clinical use of mesenchymal stem cell in the regenerative medicine field, we still struggled about the scale-up process. Since the production of the cell product needs a lot of labor, we must using high-level manpower, like master or PhD degree researchers, to produce all the process. In multidiscipline integration team, start to design an automation system, replace the human labor process to help to make sure all the process be standard by the automation system and be produced 24 hours a day to efficiently scale up all the cells to fulfill the clinical use. The automation system has several benefits. The first one is, as I have mentioned, that reduce the manpower, and actually it can reduce a lot of cost. And the second, it can reduce the contamination risk, because in the environment of production, most of the contamination risk is come from processing human. And the third one, it can standardize all the operation process. By manpower operation, it have personal bias of each one's handling. So by standardized by the automation system, it can standardize all the process to make sure a standard and specific spec. And also we have a bioreactor system to scale up a single clone of the cells. These kinds of bioreactor, the core technology of this kind of bioreactor is the matrix design. We have very efficient matrix to produce the cell, especially the harvest processing. We have fiber system to make sure the mesenchymal stem cell attachment. Then when we need to harvest the cell, the fiber system can be shake and twist to help we reduce the trypsin use and have a high efficiency of cell harvest, and the yield will be good through this process. Even by compare with traditional porous scaffold or beads matrix. At the third part, I will introduce our tissue printing system about the skin tissue printing. In E3, we have an epi-tree for thickness skin tissue construct through traditional process of tissue culturing and differentiation. Then, in order to standardize all the process, the multidiscipline team of E3 think about printing and automation may be the solution for the production of the skin. Then we design the printing module and the automation system, and also the image controlling of the printing process. We also develop bio ink that enable the tissue differentiation, and also have a cell ink that combine with different kinds of cell. In tissue, it's keratinocyte or fibroblast to make sure their activity and to make sure their survival. And also the gene expression to differentiate to mature skin tissue. So in this system, we can see the fully integrated technology of different kinds of field, like a mechanical field, like light and ICT field, and also biomaterial and chemical field, combined with molecular biology and cell biology. We have now a very good spec of skin tissue processing, and the bio ink and also cell ink are now cooperation of different kinds, different companies in the different area of the world. Here shows the printing of full thickness skin tissue. We have 3 modules of printing system to make sure the dermal layer of printing and the epidermal layer of printing. And now this system make the first in class in the world that the most efficient differentiation of skin tissue only in 6 days. It can fully differentiate to functional with barrier function skin tissue in dish. And now we can use this to test the cosmetic product and the chemical product, and also some transducible drug development and also medical device development.